we want to have an impact on India, we need to understand Indians. There's so little information on aging on the Indian population. And I also take the point that there's not one thing as in someone who's an Indian. There are different regions.
Exactly. Yes. By the way, I think genetically recently discovered we are the most mixed ethnic population in the world. So I think where we are in India is a little bit going towards that, since I stay in both places, I can compare. We're going more towards that Malay diet and slightly higher fried, high carb, like the Asian version of the bad American diet.
Yeah.
Well, it's like,
it's funny because you think of India, you think of so many vegetarians, and I go around the world telling people, one of the healthy diets you can adopt is something that's very close to vegetarian, low dairy, low red meat, lots of green leafy vegetables.
welcome to the Biopeak Live podcast brought to you by Longevity India. Today we have Brian Kennedy with us and my co-host Deepak Saini.
Hello, welcome to one more session on Biopeak Live, and it's an absolute pleasure to have Brian here. Brian, you are visiting us, and we are very thankful to you that you're coming all the way from Singapore, which is not such a long distance away from us, and coming to the beautiful, nice air of Bangalore. And I hope you have had a wonderful stay till now.
I really did. And the problem you have is that you treat me so well, then I don't need.
I would love to have you here. But before we go into the details of the podcast, let me introduce quickly Brian. Brian is technically a stalwart in longevity sphere. I mean, you've been an academician. You are currently a distinguished professor of biochemistry and physiology, which pretty much covers all aspects of longevity and health. Everything at the Luoyang School of Medicine at National University of Singapore. He's also currently the director of Healthy Longevity Translational Research Program, an Asian Center for Reproductive Longevity and Equality. That's got a lot of things built into one nomenclature. Yes, and we're hoping that you will unpack that when we go along with the conversation with you. Thank you, Brian, for taking time. And we're looking forward to a next hour of wonderful discussion with you. Prashant, you would like to start with?
Yeah, I'll start. Just talking about Singapore and the idea of blue zones, right? And there's this new kind of talk on Singapore being the other blue zone.
Yeah, the manufacturer.
Yeah, the manufacturer. If there are elements in that ecosystem that have kind of led to that moniker, what are a couple of those?
Well, I think a lot of it has been just good urban planning and an effort to care about the welfare of the population. So, you've been to Singapore, I'm sure. There's wonderful green space in Singapore. It's easy to go to parks. It's easy to, everything operates smoothly. The trains are on time. It's just everything is efficient and organized in a way that people can get through, work hard, and still have time.
Right.
So
movement is prioritized.
It's not so much trying to slow the aging process biologically. They're getting interested in that now, but it's more, how do we set up an environment that's healthy for people to live in?
Yeah. What about diet and food? Were there elements in that society that were like reasonably?
Yeah, I'd say it's mostly a good story in Singapore. I mean, I come from the United States. Anything looks like a healthy diet compared to that. You have to really break Singapore down into parts too, because there's a Chinese population, which is about 70%, and a Malay population, and an Indian population, and about 5% of potpourri. And so then we've looked at biologic age, for instance, across those different ethnicities, and the Chinese population is biologically younger than Indian and Malay. And that could be partially socioeconomic, yet it's also probably related to diet. I think the Chinese diet is...
And I think Malay diet is also a little bit...
Malay diet is tastes great, but is not the healthiest thing in the world.
So I think where we are in India is a little bit going towards that, since I stay in both places, I can compare. We're going more towards that Malay diet and slightly higher fried, high carb, like the Asian version of the bad American diet.
Yeah.
Well, it's like,
it's funny because you think of India, you think of so many vegetarians, and I go around the world telling people, one of the healthy diets you can adopt is something that's very close to vegetarian, low dairy, low red meat, not just green leafy vegetables. But the version of vegetarianism here is not the healthiest version. I have to say, I went to get dosa the other night. It was great, but I almost ask if it comes with Lipitor.
Brian, interesting point. I mean, you have transitioned from so many different spots in your career, from a pure academician coming from Bach Institute to NUS, then doing some fantastic translational research, and now setting up institutes or large-scale programs revolving around health and longevity. How is your journey for you? What have you learned? What have you decided?
Is institution building like now become the thing that really drives you the most?
I actually probably I would say no. I do that because it's a means to an end. I really want to make an impact in longevity and healthy longevity. I think that one thing that's a little bit unique about me is that I don't really want to know what I'm doing in a year. I like the idea of not knowing where my career is going and being open to new ideas. And, I've taken on leadership of institutions because I felt like it was a way to help promote the healthy longevity. And I still think that's my ultimate goal. When I started working in yeast cells, I mean, it was a long way from doing anything in humans. And in fact, people said you'll never learn anything about human aging from yeast. And I kind of believed them at the time, but it turns out we discovered sirtuins and then mTOR pathway and everything has yeast. But as things moved along, and I would have said at that time as a grad student, I just want to understand aging. I don't care about intervening or doing anything for humans. I want to understand the biology. But as I've gotten older, maybe because I'm getting older, I've come to the realization that I want, whenever my career ends, I want to be able to say some of the research I contributed to actually had an impact.
Right.
And so that has changed the equation. Now it means, how do we test things in humans? How do we measure aging in human? How do we validate interventions? How do we show benefits of health span? And so it's really changed how I think.
Well, that's pretty impressive. But what you said, something which caught my attention quite nicely, that you don't plan tomorrow. That's completely the anti-grail of longevity. You want to know what's happening tomorrow. So it's fascinating. How do you balance these two thought processes yourself?
I don't know. I think that's just my mindset. I tell everybody that I'm immortal. And it's not because I really think I'm immortal. It's just I like the behavioral patterns it creates and the mindset it creates when I believe that way. I don't worry about aging.
So before we get to a specific supplement or any supplements, one of the things we are trying to do in Longevity India is kind of get to that point where try to understand if we need to do a lot of these validation of therapies, how do we need to start to design the approaches for that? How do we, because we do have good relationships now. There's enough relationships with the hospitals and industry and all that is in place. Now we just need to know, again, technically I've learned a little bit of this. I don't come from, but how do you power them in the right way?
The first thing is, when somebody says I'm not a scientist, I've just learned a little bit here and there, it's you're in a dangerous situation because they know a lot.
So just, I know that you kind of tried to do a little bit of this, these trials in Singapore now, and because it's useful and different for this genetic population. So we want to replicate some of that in India and start that process. So how did you go about thinking about doing this in Singapore?
I still think it's a work in progress. I don't think anyone anywhere has really figured out the best way to validate longevity interventions. I'm happy to talk about that. I think the first thing here is, if you want to have an impact on India, we need to understand Indians. There's so little information on aging on the Indian population. And I also take the point that there's not one thing as someone who's an Indian. There are different regions.
Exactly. By the way, I think genetically recently discovered we are the most mixed ethnic population in the world. In terms of racial integration.
So I think, first of all, measuring aging in the Indian population, particularly if there's longitudinal data, that kind of thing is going to be very valuable because there will be differences. I think fundamentally we all age the same way, but there will be differences that could be relevant to which interventions work in this population. I think, I've just been asked to write a review on how to do clinical trials in the aging space. And so I've been starting to think about this. And
I think so. I should look at it when you're done with that. Hey, we're very useful.
Yeah, it reminds me a little bit of what you said about how to develop healthcare in India. That this don't try to replicate what's not working that well. This try to go around and build something new and progressive. And I think that's what we need to do with clinical trials and longevity too. This don't replicate disease trials.
Yes.
But let's think creatively about how we do this. And so, especially in this world we live in right now, because there's all this community data that's available. What do we make of that? Sure, it's not as good as a placebo-controlled, double-blinded trial. But it's still valuable information. Longevity clinics are doing all kinds of different things. If we can get them to baseline their baseline, do follow-ups, we can get information from that. So there are all kinds of sources of information in the longevity space that aren't available when you're trying to treat leukemia. So I think that we need to take advantage of that. We still need the sort of hardcore controlled trials sometimes for validation, but maybe we want to test multiple interventions at once and how they work together. Maybe we need to do different kinds of nested trial designs to begin to address that. So I'm giving you the questions and not really the answers here.
No, but it's good that you're thinking about it. Yeah, he also thinks
I
about
think
it.
this fascinating thing you mentioned about getting the baseline data. You were also part of the ABIOS study.
Yeah.
We'd like to hear, because from perception from India that Singapore, you already know the data.
Yeah.
Why do you need to start something which is at that level that you need to collect healthy data? I mean, because we're doing it in India, we don't have data. BHARAT study, we're doing it in India. But what is the motivation of ABIOS?
Well, there's longer longitudinal studies in Singapore, but they're almost all for Chinese population. That's one issue. So you don't learn about the other two populations. And they didn't collect data at a deep enough level for us to really assess aging. We needed omics data. We had microbiome data. And we could go much deeper in this ABIOS study. So we're really excited. We're just analyzing data now. So it's I think it'll produce a lot of interesting information. Yeah. I wish we had more people because we only had about 400 people in that study because we did so much on each one of them.
So how many are we collecting here?
We are aiming for 5,000.
5,000.
Yeah. So I think that's great. And the next step is to really think creatively how to analyze that data. Yeah. So because it's still in statistics in the biomedical space, people are doing sort of 19th century statistics on 21st century data.
Data set, yeah.
We really need to be thinking about how to model this data, use AI, be creative about what the associations it's telling us, the connections, because I view analyzing data as discovery, not as validating.
Absolutely.
And we need to take that approach when it comes to longevity. If you have 5,000 subjects, that's exciting.
No, and this is a very important point that you make, that leveraging AI not just for pure analysis but for discovery. Right? Are you going to discover novel associations and correlations?
Yeah. And so half of my lab is dry lab now, and I just sit by computers, and we've hired like a whole bunch of Russian bioinformaticians basically, and so.
Well, that's very fascinating what you mentioned, because this is something which is what Prashant and I have been talking here, that unless you have baseline. You got baseline, you got to use tools of AI to define every individual's trajectory based on a lot of baseline data. And if you collect a lot of baseline data, the more accurate the AI model would be.
I'll give you an example. I don't think everyone is going to respond to any single intervention. If it's exercise,
yes,
some level of exercise might help everybody. But if I tell everybody you need to run 3 10K runs a week, that's going to be good for some people and bad for other people.
Yes.
Any intervention is like that. So we need to be able to understand who responds and who doesn't respond.
Absolutely.
We're never going to get to personalization unless we start to stratify response like that. And right now, there's still not much data on that.
So I'm still keeping the best for the last supplements. We'll get there. But before that, your thoughts on conscious medicine. And I know that Singapore, there's been a few like Chi Health and Vishandri has started in a few of these. But in your mind, how much of this underlying science of this data on trajectory and being able to be predictive based on this, I mean, you can do a lot of omics, but if you can't leverage that, there's no use, right? So is there a little bit of a chicken and egg here?
It is. I think there is value, by the way, getting this information. It definitely teaches us. Like I've talked about a clinical aging clock. we can not only predict a person's age, but we can tell what trajectories they're heading down that are problematic, and most of those trajectories are actionable. So we can prescribe drugs or do something. So I do think we're heading in the right direction. But right now, I work with a lot of clinics, and I tell them that personalization comes through iteration. Right now, it doesn't come through some magic, algorithm that take the baseline data. We start people on a trajectory and then see where they go, and then course correct.
And that becomes a heuristic later.
Yeah, and then eventually you've got very individualized strategies for each person, but you didn't know that at the start. And that's unfortunately where they...
Have you seen any good implementations of a digital twin model where you can run these simulations and kind of extrapolate these?
I haven't seen that yet. I know lots of people are trying to create that. I think it's a great idea, but I haven't seen one really working in the longevity space yet. But I think that's where it needs to go. And in a way, we can sort of do that with our data. So we have 100,000 people that
you can do simulation.
So we can take somebody and sort of place them around the nearest neighbors and then look at what those people were doing. Some were taking this drug, some were taking that drug, and then what happened to their mortality. So in a sense, we can begin to do that, but we're not at the level you would like to be.
So I think it's about variable mapping and variables fit into the closest.
This is something that we are trying to understand. One of the startups that I'm involved with. How important is molecular pathway trajectory mapping and looking at hormonal and other metabolites at a very different level than what clinically people have been looking for, to be able to really uncover predictive pathways.
I think it's hugely important, especially hormones, but also metabolites. And we have a paper that's coming out soon on trying to measure metabolomic changes in mice with aging. And we can make aging clocks from every tissue. And one of the refreshing components of it is that some of the metabolites we already think affect aging are coming out as predictive in the clocks. So AKG is one of the main things that predicts the age of mice. So that gives us sort of closes the loop. So I think it's important. The challenge with metabolomics right now, and all omics to some extent, but metabolomics for sure, is when one lab does it here and another lab does it there.
The fundamental technical challenge on that.
It's not scalable at the moment. It's not, especially to consumers. So I like all of these omics, and even for use in consumers. The problem is that outside of genomics, which is pretty stable, everything else is user dependent and sample dependent. And
so you handle the sample.
How you handle it. So I think that the accuracy of these methylation clocks for the consumer right now is not very high. If we do it in a laboratory, we collect all the samples the same way, we store them all the same way, we run them as one batch.
Yes.
And then we get a lot of consistency, but you can't, I don't blame the companies. It's just, I don't know how they can surmount this reproducibility challenge yet. It's not there yet.
So it's not ready for
technical challenge. Yeah. So that's interesting. Let me ask the same question, because you talk about metabolome, how about running a hormone panel, a very targeted hormone? You've been doing a lot of studies with hormones. Would you put your money on that?
Yes, I would like to know that if I were an individual who's aging, especially as a woman, looking at female reproductive hormones and menopause and downstream effects of menopause. But generally, just you could list a small set of hormones I think everyone should probably measure.
So the next set of right now, that all the existing diagnostics market is, getting the blood serum, sending to a lab, do a routine ELISA to that.
Yeah.
So do you think that there would be a shift in the thought process of this by running targeted metabolomic panel?
I think that's good. I would also like to know vitamin levels for people. I mean, I can't tell you how many times somebody says, should I take vitamin D? And I say, well, what's your vitamin D level? And they're like, you can measure that? They don't even measure it. Nobody measures it. So if we knew, I feel like the vitamin and whole thing has been miscast. I think the problem is we give people multivitamins, or we give supplement with one vitamin. Even in the clinical trials, they rarely measure the vitamin levels at the start and after the intervention. And then what it's doing is you have a spectrum of people for any particular vitamin, and then you give them some supplement vitamin, and it it creates an even bigger
Spectrum.
Some people are in the optimal range. They're moved into the optimal range. Some people are moved out of the optimal range. And then the net effect when you do the meta-analysis is nothing. I think that, and we have some mouse data for this, if you could optimize all of the vitamins, you would have a big impact on aging just by doing that in the human population. But companies aren't designed to do that. They're not, the company that sells the vitamin is not designed to take blood samples
And test it.
measure. And so the economic model doesn't fit that. And so we're misusing the vitamins and not getting the impact out of it.
Oh, that's fantastic. So, Brian, there's one interesting point which I want to ask. There's something which I've always been curious about. Supplementation. We always talk about supplementing vitamin D, vitamin B12, and a whole bunch of things here. The way our body's been operating as a machine, we have a microbiome which makes these vitamins. And you take these supplements also. Every time you supplement, you take a supplement, you're actually asking your gut to make something lesser because you're absorbing it synthetically.
Yeah.
What's the impact of...
They're also selecting for some bacteria over others that
Yes.
can use whatever you're...
Exactly. So do you think such type of therapeutic intervention, whether it is hormonal or like HRT therapy or vitamins or supplements, actually, for lack of a better word, accelerating our aging trajectories because we are now tuning our system to absorb easily available resources which our body was supposed to produce?
Yeah, I think that in some cases that's a concern. I mean, you think of it this way. Anytime you perturb the system, you're changing the state of the network of the system. And so that particular intervention might have worked in that old system, but when you alter the system to a new system, it's going to be a different overlapping set of interventions that work there. And so it's kind of a moving goalpost.
Exactly.
And I think that does create some challenges. Yeah.
Because a lot of them have this feedback inhibition, and you actually fire up the system too much, it starts shutting down certain physiological processes.
But it's very independent. It's very dependent on what you're talking about, though. I think for HRT, it's not used widely enough. I think there are reasons why women should not do hormone replacement, but for the majority of the cases, I think the benefits way outweighs. So, there were studies out that were not really designed in the right way that suggested that women shouldn't do HRT, and a whole generation of women didn't do it. And we're seeing all kinds of age-related complications. And on top of that, we talk about lifespan being longer in women, but health span is not necessarily longer. It is. And that's probably due to a drop-off in hormones. So I think that that's relatively low-hanging fruit if we want to do something about aging in a way.
Coming to some of the supplements you worked with, right? So, was Spermidine a very early one that you picked up?
Yeah, we did some work on Spermidine. We actually, sometimes in my lab, we do things that I think are not the best things for publishing in Cell Science and Nature papers because we spend a lot of time validating what other people do.
Right.
And I do that for a couple reasons. It's gotten me in some big fights in the past, but I do it for a couple reasons because one is often we're looking at taking those things into human clinical studies. Right? And I feel like if somebody has shown something in a mouse, if I can repeat it in a mouse in my lab, it's much more robust result.
Yeah. Right.
If I can't repeat it, it doesn't mean that something bad was done on the other side. The conditions were different. Who knows what happened? But if I can repeat it, I believe it. And so that's one reason we do it is that you could spend a little bit of money in mice or a lot of money in humans. I'd rather optimize the chances it's going to work. Spermidine is one of those supplements that shows really promising data. We saw improvements in metabolism with aging. And we saw the mice lived longer too. That was reported by a different group, but we were able to confirm that. And so I think it's a very good candidate right now.
And human trials post that?
We're not doing it, but there are human trials underway with Spermidine.
So these are areas where we can pick up.
I think there are a list of things that would be interesting to try. Spermidine is one of them. Urolithin.
You should do ACG and
AKG. I'm biased there. But
I know. He biased that.
And a few other things that look very promising right now. I think astaxanthin looks promising.
Yes.
And I think there are probably a lot of things that come from Ayurveda that are promising too. I don't know them very well, but I suspect.
Yeah, see, of course, Ashwagandha is now well known.
Ashwagandha, yeah.
And
yes, Brahmis.
Berberine.
Berberine, yes.
Brahmi.
Yeah, I don't know.
Brahmi
is like a extract which is used improves neural cognitive function, and it is something which has been shown time and again in all kind of animal trials. But the interesting bit of Brahmi is it works in a lipid form.
Oh, I see.
So it is not a water soluble. It comes in we call it the grita, basically the lipid extract form. It seems to work way better.
It's memory, a little bit of memory.
Memory, cognition, late stage dementia. It seems to help in a lot of different things.
So I would say, testing some of the things that come externally, but also maybe some of the internal things would be interesting here.
Yeah. So one of the interesting thing which is like, a lot of therapeutic interventions. So you do trials in mouse and then you take them to human. So, I mean, if you just on top of your head, if you have to say 5 molecules which worked in mouse very well and should be done in human, and 5 which didn't work in human but worked in mouse, or maybe 2, 3, if not 5.
I think you want me to give specific examples. I mean,
Love to know them.
we don't have a lot of human data yet.
Yeah.
And that's the challenge.
That's the part.
Yes, yeah.
I'm willing to sort of go on a limb and say that the correspondence between mouse aging studies and human aging studies is going to be bigger than mouse disease and human disease studies. Okay, there's a fundamental difference here, because when you try to study a disease in a mouse like Alzheimer's, well, they don't get classic Alzheimer's. So now you have to engineer a disease into a mouse. You put human genes in and mutations. And then when you do that, you get a 6-month-old mouse that gets a disease. But that's a young mouse. And it's old humans that are getting that. So you've changed 2 major parameters, and then you find things that work in the mice and they don't work in the humans.
Yeah, so that's why a lot of these drug therapies.
That's why I think a lot of them don't work. First of all, people should use age-appropriate mice. But when you talk to a mouse scientist, they say, "Oh, it costs less." One thing about the human trial that fails, how much that cost?
Yeah.
But that's not the mindset.
Yeah. That's interesting. So what you're saying is, unlike disease model system where there's a lot of fear, aging studied in mouse is going to be much more
aging in a mouse versus natural.
That's an interesting.
There are different features. They're not identical, but I think the core pathways are going to be the same. I think that's what we've learned, because that knocking down mTOR signaling in a yeast extends lifespan all the way up to, I think, humans. So and all of the conservation. Yeah,
conservation. So, and if you had to look at combination therapy on this natural supplementation, like the trim one, and any are more promising for you than others?
I think that's an interesting one. Right now, N equals 1, but I'm taking sublingual NAD, and I think with AKG that works really well together for me.
So, and this is against you've tried NR and you've tried niacinamide.
There's a company in Australia and Singapore that has a strategy for sublingual delivery of it. Interesting. And I noticed something with that, and that's one thing I encourage people to do. If they want to be on the cutting edge and try supplements, I think that's fine. But pay attention to your biology. And first of all, measure things before you start. Second of all, if you like, what I noticed when I did the NAD with the AKG is that it uncouples my heart rate from my respiration when I exercise. So I'm running. I do a lot of running, and I'm running. My heart rate's going up normally, but my respiration's staying less, and so I'm running faster and more efficiently.
So it's in a way your VO2 is better. Is that?
It's either VO2 or maybe just your vasculature is delivering oxygen better. There's something that's happening there that's different, and so if you want to be on the cutting edge, don't just take things. You pay attention to what's happening to your body, because you're going to notice differences in sleep. Some of it's very subjective. You might think that you're cognitively more active, and it's hard to deal with that. But I think people don't just take stuff. Just so pay attention.
On AKG, I know you have a bias there, but over time, are you seeing more validation there? Has it been just the same levels? Where do you see the?
Well, we're looking at data right now from a methylation company where they just took 5,000 samples they ran, and they did questionnaires with people, what they were taking, what their medical stuff, and they ranked the supplements, and the Rejuven, which is the one that I helped develop, that comes out. It has a time release AKG, came out number 1 in that list. So we're going back and like really deeply analyzing that data and trying to see. So we're doing a clinical trial that's controlled, double-blinded in Singapore as well with Andrea Maya. We're just getting that data back, and so we'll see what that says.
How big was the cohort?
That was about 120 people, I think we did. And
Interesting. That's interesting. So we could.
but that's just the time release AKG, not the other stuff that's in Rejuven. But it's we're gonna see. I think for any of these things, we don't know the perfect trial designs yet. We're doing it in different populations. Some things may work here and not there.
What were you looking at as
Indicators.
for the AKG?
So when we do the studies in Singapore, we have a primary indicator. In this case, we chose biologic age using methylation class. But we're using a whole set of other parameters. We're measuring inflammatory factors. We're measuring pulse wave velocity. We're measuring grip strength, and we're measuring all kinds of other measures of metabolism. So
3 months, 2 months.
we get 6 months intervention, a 3-month follow-up. Because you want to know if you stop taking it for 3 months, do things go back?
Right.
And what was the duration of recording? Every month or every?
I think they made an initial visit, intermediate visit, 6-month visit, and then that.
All right. Makes sense. That's interesting. So basically, what you're saying, there's a lot of merit in doing, given that we do not know which way it's going to get. What there's never any number is a good number than no number.
Yeah, but we're still learning. I can't tell you that's the perfect strategy. So other people would say you need to do like the TAME trial and give 3,000 people metformin and see if you could prevent disease. That's a great trial, but it costs a fortune.
Exactly.
And so we're all learning.
It's all for the next generation.
Yes. Yeah. You talk about measurements. I need to put. So if we heard a lot about different type of clocks, this clock, that clock, Grimm clock, inflame clock, where would you bet your money on?
The clock that's not made yet. This is still research.
And that's
We're
what
still improving the clock as we go. And they're great tools, and some of them are they're useful in populations. I think some of them, if they're done right, are useful in intervention studies at the individual level, but none of them are perfect yet. And my guess is that they're measuring overlapping features of aging.
Yeah.
So if you think of aging as hallmarks, some are more balanced toward one and some are more tipped toward other.
So there's a skew.
Yeah. I like the second generation clocks that predict outcomes, though. The first generation clocks, when we compared our clinical chemistry clock to the methylation clocks, some of the first generation clocks were worse than chronologic age at predicting mortality. That's a bad sign. Is your passport better at predicting when you're going to be alive in 200 months? There's a problem.
Yeah.
But most of the second generation clocks have some predictivity, which is, and so that's good, but there's a lot of improvement that can still happen.
What about clocks like Dunedin pace, right, which is more showing the pace of aging and so on?
Dunedin is really interesting. It is measuring the speedometer in the car, the odometer.
And seems to have more data and more, I don't know, consistency.
Yeah, we use it. I like it. And I think it's still early with that one because it's the only one I've seen that really tries to measure that
speedometer.
Yeah, and so there are probably ways to improve that too. But I think it's been a useful addition to the field for sure.
It's just wait and watch, as you rightly say.
I have a good score on that one, so I like it.
Yeah, that's a good one. So some quick quiet questions.
Yeah, I think we covered most of the topics. I think maybe if there were one or two things that I asked Matt also this question yesterday.
Yeah.
We are right now looks like there is a deficit of fundamental unlocks.
Yeah.
So your group and what you did in the mTOR unlocks and some of those were like you said, they then led to a whole host of other downstream understanding of aging. Do you believe that there is one or two promising areas where this unlock can come from? And is inflammation, some research around inflammation that will unlock something, is which in any areas, one or two areas?
Well, first of all, anything Matt said was wrong. So I'm just saying that.
No, that is, we had some nice discussions with Matt. When you see our part,
we have like 70 papers together, so we tease each other a lot. But I think we think very similarly, though. Well, first of all, I would still say the mTOR pathway is extremely promising. I think that the rapamycin, if I had to pick one drug, I'd still pick that one. I've taken it, a lot of We were estimating that up to 100,000 people are taking it off.
So do you do the weekly?
When I do it, I don't do it all the time, but when I do it, I do once weekly. Yeah,
Once weekly, and you cycle it every 6 months, every 3
I
months?
try to do like 10 weeks, but once a year or something like that. And part of the reason for that is I'm trying many different things, but I don't like trying many different things at the same.
Which is very,
Yeah, so I have AKG all the time, but then I add one thing.
you add one thing and just see the.
But I think that's important. Everything we do keeps coming back to inflammation. You know, when we built this clinical aging clock, the primary predictor of aging were basically inflammatory.
Yeah, so that there
Yeah,
could be some correlations AI can unlock that I don't.
yeah, I think so. Inflammation is always interesting. There's data on IL-11 recently that's
Yes.
I think reprogramming is very exciting. I think senescence is a bit overblown, but I think reprogramming is very exciting.
Senescence, even Deepak strongly believes that.
I don't want to. I mean, I think senescence is certainly a driver of aging, but it's like a pendulum. You know, they go too far in one direction. And for a while, everybody said senescence is not.
We just have to kill. Yeah, that's where your senility said.
And then we go too far in the other direction.
And then it drives everything.
Yeah, I think the challenge of senescence is that finding a drug that effectively kills senescent cells and doesn't have off-target effects that are toxic.
It's not possible.
Yeah.
It's not possible. As like, how much, when, and which one.
Yeah. Certainly for age-related diseases, it would be useful. And if you had a good senolytic, it would extend lifespan.
Yeah.
But
and I think senolytics are very good application in cancer therapy. Yes, because it's a fantastic
and also like, COPD, lung diseases. So I'm not saying it's a bad thing. I just think we got too excited for a bit. Yeah. I think that reprogramming is really interesting because there's some data that's really hard to describe from our Gerophysics conference last week that suggests that the interventions we're doing now are going to square the curve. So they may give you an average of 5 or 10 years extra median lifespan. And longer health span, which is, by the way, a revolution if it happens.
Yes.
But as they're not going to affect maximum lifespan. And to do that, you need to do something else. And one way to deal with entropy is replacing cells. Yeah. And so reprogramming has some really interesting possibilities. It's not ready for prime time yet.
Exactly.
What about stem cell therapies? Do you think they are kind of just transitory in terms of their effects, or do they really persist and kind of stay over a period of time?
I think they're also very promising. They're probably in that moderate benefit side, level, but I think they're very promising. And one of the challenges is there's a whole regenerative medicine field and a longevity field, and they don't talk to each other.
Yes.
And so we need to bring that closer. I've worked with stem cell clinics. I've tried MSCs. I've had MSCs IV. And, I think that it's an interesting area. Unfortunately, there's partly going back to George Bush stopping stem cell research in the U.S. It's pushed stem cell research into these gray areas. You don't have great clinical trials sometimes on MSCs or ADSCs or any of this stuff. But I think there's certainly a good reason why they might work. They're anti-inflammatory. They persist in the body for a few weeks, and they can have a long-term effect on inflammation. They may stimulate your adult stem cells to function better.
Yeah.
All of that's very promising, but we need better data on it.
So
Peptides are really interesting, I think. I'll give you an anecdote. I spent 2 days with this terrible viral stomach thing last week, and I was like not getting out of bed, not moving, muscle aches, and I happened to be texting a functional medicine doctor, and he said, "Open your door in 30 minutes and take it." And so I went to the door. In 30 minutes, there was an envelope on my. And it had a syringe in it that said, "Take me."
As simple as that.
Well, it turned out to have 3 peptides in it that are supposed to be anti-inflammatory. And so at that point, I was like, well, dying's not the worst thing at this point. So I just took it.
Yeah.
And within 4 or 5 hours, I had less inflammation, less fever, less muscle aches. It didn't cure the virus, but it made me
feel better.
Treated the symptoms,
So peptides, I think they're very promising.
They're very promising. I don't know enough. And that's again a gray area regulatory one.
Yeah, it's yes.
I think certainly peptides are active, and if people are willing to do these self-injections, which are not that problematic, then
So it looks like the side effects aren't a lot crazy when it comes to peptides, right? So
we need more research. I think a lot of difficult to say. Yes. Yeah, but it's I don't know. try to pay attention. I don't think I know enough about a lot of these peptides, and most of the longevity field is kind of ignoring them.
So just you're saying that in all these areas you have this regenerative medicine and stuff. There's just no collaboration and convergence.
We need more collaboration. Yeah, and yeah, the peptide space is interesting. It's like, where do you get the peptides made? It's like American companies. The FDA's kind of come down against peptides, so now you can't get them produced. Then you're getting them made somewhere in Russia. I mean, that could be fine, but I don't know where the
make it. Yeah.
is or anything. And so it's I'd rather bring things into the light, and honestly, I think clinics are doing that, and the ones that are doing it the right way, I'm happy to work with, because I think that I think we need to empower people. Okay.
To make decisions.
And what's happened is that doctors have taken too many decisions out of the hands of the patients. And so that's why there's low compliance on drugs. Like I say, your cholesterol's a bit high. Here's some statins, stink dash. Half the people don't take it, because they're not invested in that. They don't really know what that means. Maybe they have heart risk down. Yeah. You need to invest people. That means educating them and then letting them make choices. And if they want to be like on the edge trying things, and they know the benefits and potential benefits, they shouldn't be on risk. Fine. I'm all for hackers. I don't agree with a lot of what some of them are doing, but I think they have the right to do it.
Right.
And if we can invest people in making their own decisions more, then I think we'll get better better outcomes. And so that's why I like what the clinics are trying to do.
So we'll wrap up with last question. We started with Singapore. I'll end with Singapore.
Yeah.
What do you think we can do more between Bangalore and Singapore over time?
I think there's a lot to be learned everywhere. Just one question I think is interesting is what's going on with aging in Indian population in Singapore versus India? Because there you have similar genetics, different environment a little bit. So I think that would be interesting.
Very interesting comparative longevity analysis.
And more very nice. Maybe some trials on these.
Working together on intervention trials, I think is great. If we could do this at multi-centers and work together, and so we're happy to provide our 2 cents on trial design or anything like that. Happy to collaborate.
So we'll really forward to maybe next time we talk, we'll talk on some trial data.
Good stuff.
Thank you, Brahma.